We'll tie this all together in a breast cancer model with Dr. Andy Dannenberg.
Okay, well, first, thank you very much to the organizers for including me in this very interesting meeting. And as you can see, the title of the talk is Breast Adipose Inflammation, a Silent Killer. Obesity is a risk factor for the development of hormone receptor-positive breast cancer in postmenopausal women. It's also a poor prognostic factor for breast cancer patients. This is a schematic illustrating some of the pathways linking obesity with breast cancer. In the obese, one sees increased estrogen synthesis, which can drive the development and progression of ER-positive breast cancer. Insulin resistance also occurs in the obese, and deregulated insulin signaling can also contribute to the development and progression of breast cancer. And one can see altered adipokine and cytokine production, which can also contribute to the pathogenesis of breast cancer. And today what I'm going to talk about more specifically is inflammation in the adipose, which intersects with each of these pathways. So this is a cartoon demonstrating that obesity can cause an inflammatory state. The crown-like structure was first recognized in 2003 in adipose, and then we found it, as I'll show you, in breast tissue in 2011. In obesity, one sees adipocyte hypertrophy, and ultimately a cell will become large and hypoxic, and ER stress can occur, leading to a sick cell. Chemokines such as MCP1 are released, and one can see recruitment of mononuclear cells from blood, which differentiate and then become macrophages, forming this crown of macrophages around the dead or dying adipocyte, hence the term crown-like structure. These macrophages then phagocytose the dead adipocyte using a process known as lysosomal exophagy, and in the process become foam cells. In this process, one can find increased cytokine production such that the presence of crown-like structures can be associated with elevated levels of pro-inflammatory mediators. Importantly, in obesity, one also sees increased lipolysis secondary to insulin resistance, and saturated fatty acids in particular can activate macrophages, leading to a pro-inflammatory state. So during the remainder of the presentation, you'll hear me use the term crown-like structure, or CLS, or crown-like structure of the breast, or CLS-B, and that refers to this morphologic entity. So our first objective was to determine if crown-like structures of the breast exist in women, and did they correlate with BMI. To do this, breast tissue was obtained from women who underwent mastectomy. I should point out that we utilized the uninvolved quadrant of the breast in mastectomy specimens, and we performed routine H&E staining and CD68 immunohistochemistry, CD68 being a macrophage marker. And here one sees that crown-like structures of the breast are common in the breasts of overweight and obese women. This is what lesion looks like by H&E. Here you see macrophages decorating this dead or dying adipocyte, and down below you see the same lesion stained for CD68. And if anybody wants to do this, it's much easier to pick out these lesions when you do CD68 immunohistochemistry as opposed to H&E. And then when we looked at the relationship between BMI and the incidence of crown-like structures of the breast, one sees that in those who are obese, the incidence is on the order of about 90%. In the overweight, it's probably 50 to 55%, and fully a third of normal BMI women have occult breast inflammation. I'll have much more to say about the normal-sized women later in the presentation. Here we demonstrate that adipocyte size correlates with both BMI and crown-like structure of the breast status. So in panel A, one sees a nice linear relationship between BMI and mean adipocyte size in the breast. And then in panel B, one sees that the mean adipocyte size is larger in those who are inflamed versus uninflamed. So the take-home message is larger adipocytes correlate with inflammation, and we think that that's because of the combination of hypoxia and ER stress. And I'll come back to this point later on. So having discovered this process, the question then became, is it important? And so our objective was to determine whether breast white adipose tissue inflammation manifested as CLS-B is associated with shortened recurrence-free survival in women who develop metastatic breast cancer. So this is a retrospective study, and there were 127 cases, and you can see at the top, 75 of these cases had no evidence of white adipose tissue inflammation, and 52 were inflamed, CLS-B positive. And then we simply compared those without inflammation versus those with inflammation, and there are a number of interesting features. One can see that the women who were inflamed were on average older. They had, as expected, a higher body mass index. They were more likely to be postmenopausal. And interestingly, they had features of the metabolic syndrome, including hyperlipidemia, hypertension, and diabetes. That is an increased incidence. This was of great interest to us because others had previously shown that those with metabolic syndrome had a worse prognosis, and therefore we wondered whether or not inflammation might help to explain that phenomenon. Here we went on to look at the relationship between CLS-B status and distant recurrence-free survival. The gray solid line represents those women who did not have inflammation at the time of index mastectomy, and those represented by the dashed red line represent women who did have occult breast inflammation at the time of mastectomy. And one sees that the time to recurrence was reduced in those who were inflamed. More specifically, the median was 20 months in the CLS-B positive cases and 26 months in those who had no evidence of inflammation. And this was true when we controlled for all the critical variables. Take-home message is that the presence of occult breast inflammation was an independent prognostic biomarker. And since we did this work, another group has now reported that the presence of crown-like structures of the breast is associated with worse overall survival as well. So based on these findings, we concluded that breast white adipose tissue inflammation is associated with metabolic syndrome. In patients who develop metastasis, white adipose tissue inflammation is associated with shortened distant recurrence-free survival. And whether white adipose tissue inflammation promotes the progression of breast cancer via local effects, intra-breast effects, systemic mechanisms, or both is uncertain. And so consequently, that was the next question we raised. Is white adipose tissue inflammation associated with local or systemic effects that can promote breast cancer? So to address this question, we assessed CLS-B status in another 100 women with varying BMI. We performed RNA-seq on non-tumorous breast tissue in all 100 subjects, and we assessed circulating levels of CRP, IL-6, insulin, and glucose. And here you see a description of these 100 women. You'll note that 52 were CLS-B positive and 48 CLS-B negative. Once again, those who were inflamed were a bit older. They had a higher BMI. They were more likely to be postmenopausal, and there was a suggestion of features of the metabolic syndrome. And here you see the results of our RNA-seq analysis. The take-home message is that breast white adipose tissue inflammation, a histologic biomarker, if you will, is associated with numerous changes in the transcriptome. And I realize this is a mixed audience, so as a reminder, each of these columns represents a different subject. Each of these rows represents a different gene. Here you can see the CLS-B negative cases separated by this white line from the CLS-B positive cases. And the take-home message is that there are a large number of differentially expressed genes in breast tissue in association with inflammation. We went on to demonstrate that breast white adipose tissue inflammation is associated with elevated levels of CRP, IL-6, insulin, and glucose. I should point out that when you have occult breast inflammation, we've previously demonstrated that it's a sentinel for inflammation and other fat depots. So when you begin to think of this as a sentinel for what's going on diffusely in fat, it's teleologically reasonable that these blood-based biomarkers are deregulated. We then broke the code for this particular cohort, and consistent with our other work, overwhelming majority of obese patients were inflamed, and again, roughly a third of the normal BMI women were inflamed. And from my perspective, this is probably the most important slide of the talk. For many, many years, I have wondered why a normal-sized woman who was wild-type for BRCA1 or BRCA2 might develop breast cancer. That's a fundamental question. And so I hope to convince you that inflammation is likely to be contributory. So we decided to focus on these normal BMI women. And here again, we looked at adipocyte size, and white adipose tissue inflammation was associated with breast adipocyte hypertrophy in normal BMI women. So here you see the CLS-B negative normal BMI women, the CLS-B positive women, and again, we find a significant increase in adipocyte size. So they have a normal BMI, but consistent with this meeting, they are undoubtedly hyperadipose, manifested by enlarged adipocytes in the breast. Again, we performed RNA-seq, and we were able to demonstrate, even among normal BMI women, that the presence of inflammation correlated with a large number of differentially expressed genes. And one that we were particularly interested in and have been interested in for many years is CYP19A1, which encodes aromatase, which was upregulated in the CLS-B positive cases, which we then confirmed by qPCR. Just as a reminder for the group, androgens are converted by aromatase to estrogens, and aromatase is abundantly expressed in adipose tissue, particularly in adipose stromal cells, and can synthesize and synthesizes estrogen. And so consistent with our RNA-seq data and qPCR data, we were able to demonstrate that aromatase protein levels and activity are increased in the inflamed breast tissue of normal-sized women. So here you see a Western blot of 6 CLS-B positive normal BMI women and 6 CLS-B negative normal BMI women. So a clear increase in aromatase protein, that which makes estrogen, and a corresponding increase in aromatase activity. Is the aromatase functional? Well, this is a publication from a different group earlier this year, where they were able to evaluate the relationship between crown-like structures of the breast and sex steroid hormones in breast adipose tissue and serum, and they found a very nice correlation between the estrogen to androgen ratio being higher in those who were CLS-B positive, whether you looked in the breast tissue or in the serum, consistent with the increase in aromatase that we had observed. We wondered whether these normal BMI inflamed women might be metabolically obese, and so we looked at their blood. And here you see data from 72 women. Again, all of them had a normal BMI. All of them would ordinarily get a pat on their back by their primary care doc, telling them to keep up the good work because their BMI was normal. And the answer is not so fast. So take a look here. There are 28 of the 72 were CLS-B positive, and we found elevations in C-reactive protein. There was a 50% increase in leptin levels consistent with the enlarged adipocytes, which I already showed you. Triglycerides were elevated, and there was an increase in fasting insulin levels and in HOMA2IR, suggesting that they were relatively insulin resistant compared to the normal BMI uninflamed. So you then have to ask whether or not these circulating changes are important and what's their significance. A different group recently reported that elevated insulin levels are associated with increased breast cancer risk in normal weight women. So here you see the data. Everybody consistent with our study had a normal BMI, and then they were either metabolically healthy or metabolically unhealthy as defined by their insulin levels. And those who are metabolically unhealthy because they had elevated insulin levels had a doubling in their risk for breast cancer. So I would argue, based on our more recent findings, that those who are hyperinsulinemic, yes, they're at increased risk for breast cancer, but they're also likely to have occult breast inflammation and numerous other changes at the level of gene expression, such as the elevation of aromatase, which I shared with you, and multiple other blood-based abnormalities. So it's likely to be the constellation of effects that are important for increasing the risk of breast cancer. So in conclusion, white adipose tissue inflammation is associated with numerous changes in gene expression in the breast. It's associated with systemic changes that occur in the metabolic syndrome. White adipose tissue and its associated pathophysiology occur in a significant subset of normal BMI women, and this is a potentially targetable process linking obesity or excess adiposity to breast cancer. And finally, I'd like to acknowledge my colleagues. We've worked together as a collaboration for many, many years, and this has been a through-and-through effort between investigators at Weill Cornell, where I'm located, and across the street at Memorial Sloan Kettering. So thank you for your attention.